Welcome to Bio Bits and Bytes.
Great to be here.
Today we're diving into something really fascinating. It's where our body's metabolism and maybe the fight against cancer could intersect,
Yeah, it's about cold exposure activating brown fat.
and how that might actually suppress tumor growth. Sounds pretty wild, right?
It does, but there's some compelling science emerging. We've got a key study, brown fat-mediated tumor suppression by cold-altered global metabolism, plus some other papers giving context.
Okay, let's dig in. The big question we want to unpack is, could just getting cold, activating this brown fat, actually be a new way to tackle cancer?
That's the core of it, a potentially novel therapeutic approach.
Seems almost too simple. But all right, let's start with the basics. Cancer metabolism.
Yeah.
We hear about the Warburg effect. Can you break that down for us?
Absolutely. So the Warburg effect, or aerobic glycolysis, is this observation that cancer cells, they metabolize glucose differently than most normal cells.
Differently how?
Well, healthy cells typically use oxygen in their mitochondria, the powerhouses, to break down glucose very efficiently for energy, ATP.
Right, standard cellular respiration.
Exactly. But cancer cells, even when there's plenty of oxygen around, they often favor a less efficient process. They
Right.
take up a lot of glucose and ferment it into lactate.
Wait, less efficient? Why on earth would they do that? Doesn't seem very smart for survival.
It seems counterintuitive, yeah. But think about what cancer cells need to do. Grow and divide fast.
Ah, okay. Speed over efficiency.
Precisely. While this rapid glycolysis makes less ATP per glucose molecule, it churns out the building blocks, the precursors for lipids, nucleotides, amino acids much more quickly. Things they need for making new cells, invading tissues, metastasizing.
So fuels rapid proliferation.
Exactly. And it's not a minor thing. Something like over 70% of human cancers show amplified genes for this glycolytic pathway. They are seriously hooked on sugar.
Okay, so cancer cells are, like you said, hooked on sugar using this fast, less efficient pathway. Now, connect that to brown fat for us. What's brown fat's role in our metabolism?
Right, so brown adipose tissue, or BAT, unlike white fat, which mostly just stores energy.
The stuff we usually complain about.
Oh, yeah. Brown fat is specialized to burn energy. Its main job is generating heat. It's called non-shivering thermogenesis.
Non-shivering, meaning it warms us up without the muscle contractions of shivering.
Exactly. It's a key way mammals stay warm in the cold.
How does it actually make heat
Yeah.
at the cellular level?
It comes down to the mitochondria again. BAT mitochondria have a special protein called uncoupling protein 1, UCP1.
UCP1, okay.
Normally, mitochondria create ATP by letting protons flow across a membrane through a specific channel, like water turning a turbine. UCP1 basically pokes holes in that membrane. It creates a shortcut for the protons.
A leak, sort
A
of.
controlled leak, yeah. So instead of that energy being captured to make ATP, it gets released directly as heat.
Fascinating. So BEAT burns fuel to make heat via UCP1. How do we switch it on?
Cold is the main trigger. When you get cold, your nervous system releases norepinephrine.
Okay.
That norepinephrine binds to receptors on the brown fat cells, kicking off a cascade that activates UCP1, and boom, heat production ramps up. Certain drugs can mimic this too.
And importantly for our discussion,
Importantly, BAT is also a major glucose consumer. To fuel this heat production, it needs energy. Studies show if you block glucose uptake or its initial breakdown in B, the thermogenesis gets impaired.
okay, so cancer loves glucose, BAT loves glucose when activated by cold. I think I see where this is going. What did the mouse study show when they put these two things together?
This is where that main study comes in. Researchers took mice with various types of implanted tumors: colorectal, fibrosarcoma, breast cancer, melanoma, even pancreatic cancer.
A real range of cancers.
Yes, and they put these tumor-bearing mice in a cold environment, 4 degrees Celsius.
That's properly cold, like a fridge.
It is, and the results were striking. Compared to mice kept at a warmer, standard lab temperature, the tumor growth in the cold mice was significantly inhibited across all those different tumor types.
Wow, significantly inhibited.
Yeah. And interestingly, they didn't see the strong effect at 22 degrees Celsius, which is kind of room temperature for mice.
So the level of cold matters.
Yeah.
Was the effect permanent? Did it kill the cells?
Good question. No, it seemed reversible. When they took the mice out of the cold, the tumor started growing again.
So it's not killing them directly, more like slowing them down, putting the brakes on.
Exactly. And they confirmed this by looking at the tumor cell cycle.
The stages of cell division.
Right. In the cold-exposed mice, more tumor cells were stuck in the resting phase, G0, G1, and fewer were in the active phases of DNA replication, S phase, and division, G2M phase, so proliferation was definitely hindered.
And did this translate to better outcomes for the mice?
It absolutely did. For mice with colorectal cancer, acclimatizing them to the cold almost doubled their survival time compared to the warm group.
Doubled survival.
Yes.
That's huge in preclinical models.
It really is. And they also found that using a drug, one that specifically activates those same beta-3 receptors on brown fat that cold activates, had similar tumor inhibiting effects.
Okay, that strengthens the link to B activation.
Uh,
So the cold slows tumor growth, extends survival in mice. But what's happening inside the tumor? The microenvironment.
right, the tumor microenvironment, or TME, they looked into that too and found some, well, somewhat surprising things.
Oh, like what?
Well, for one, even though the number of blood vessels, the microvessel density, actually decreased in the colorectal tumors in the cold.
Fewer blood vessels.
Yeah,
You'd think that means less oxygen, more hypoxia.
you would think, but counterintuitively, the oxygen levels within the tumor actually improved. Hypoxia was reduced.
How does that work? Fewer pipes, but better flow. Or maybe less demand.
But that's likely the key. The reduced metabolic activity and proliferation of the tumor cells probably lowered their oxygen demand so much that even with fewer vessels, the relative oxygenation got better.
Okay, that makes sense. What else changed in the TME?
They saw that substantial inhibition of tumor cell proliferation using a marker called Ki-67.
Ki-67, right? Marks actively dividing cells.
Exactly. Lower Ki-67 means less division. They also saw a decrease in certain immune cells, these CD45 plus myeloid cells.
Myeloid cells, a type of immune cell, often involved in inflammation or suppression in tumors.
Can be, yes. Though, interestingly, they didn't see big changes in overall cell death apoptosis or in other immune cells like macrophages or fibroblasts.
Okay. And these TME effects, were they consistent?
Yes, pretty consistent across different tumor types and locations, whether subcutaneous or in the liver. And crucially, they measured core body temperature and the temperature inside the tumors.
Did the tumors get cold?
Nope.
Hmm.
Largely unchanged, which really argues against the idea that the cold is just directly chilling the tumor cells into submission.
Right. So it's definitely a systemic effect triggered by the cold environment, mediated by something else in the body.
Exactly, which leads us squarely back to brown fat.
How did they prove BAT was the crucial mediator?
This was the really elegant part of the study. First, they confirmed the metabolic shift. Cold exposure boosted overall metabolism in the mice and caused a significant drop in blood glucose levels.
Lower blood sugar.
Mm.
Makes sense if BAT is burning it.
And improved insulin sensitivity, too, clearing glucose faster. But the clincher was the BAT removal experiment.
They surgically removed the brown fat.
Yep. In mice, without their main BAT depots, exposing them to cold no longer suppressed tumor growth.
Wow. Okay. That's pretty definitive.
It is. And those beneficial changes in the TME, also gone in the BAT-removed mice.
So BAT is absolutely necessary for the anti-tumor effect of cold.
And they visualize this using PTC2 scans, tracking that radioactive glucose analog, 18F-FDG.
Right, to see where glucose is being used.
In normal mice in the cold, you see bright spots light up in the BAT regions, high glucose uptake. In the BAT-removed mice, nothing lit up there in the cold. And importantly, the glucose uptake in the tumors of these BAT-less mice stayed high, even in the cold.
So removing BA prevented the cold from activating that glucose sink, and the tumor kept getting its fuel.
Exactly. The cold activates BD. BD gobbles up glucose, and the tumor, which relies heavily on glucose, gets starved. That seems to be the central mechanism.
Okay, beat activation is key. How do they show the metabolic reprogramming link more directly? The shift in fuel use.
They used some powerful tools. RNA sequencing to see gene activity, and metabolomics to measure the levels of small molecules, the metabolites.
What did those tell them?
In the tumors of cold-exposed mice, genes involved in glycolysis and also lipid metabolism were turned down. Activity was lower.
Less sugar burning, less fat burning in the tumor.
Right. But conversely, in the brown fat of those same mice, glycolysis genes were turned up.
So beet is ramping up its sugar use while the tumor is dialing it down.
Precisely. And the metabolomics confirmed it. Key metabolites in the glycolysis pathway were lower in the tumors and higher in the beta of the cold mice.
Like the intermediates in the sugar breakdown process.
Exactly. And interestingly, the activity of LDH, the enzyme that makes lactate at the end of glycolysis, wasn't really changed in the tumors. Meaning the lower lactate levels they saw in cold-exposed tumors likely resulted from less glucose entering the pathway in the first place, not from lactate being broken down faster. Reduced production, not increased clearance.
It really points to resource competition.
Hmm.
Bait wins the glucose tug of war in the cold.
That seems to be a major part of it. And they tested this dependency on glucose availability directly.
Oh.
They gave some of the cold-exposed mice a high glucose diet, basically flooding the system with sugar.
And what happened?
The anti-tumor effect of the cold vanished. Tumors grew just fine when glucose was abundant, even with activated BT.
So the glucose restriction caused by bait activation is essential for the effect.
It appears so. And one more piece of crucial evidence involved UCP1.
That key protein for heat generation in Bt.
Right. They used genetically engineered mice that lacked the UCP1 gene, UCP1 knockout mice.
What happened when they put these mice in the cold?
Cold exposure did not inhibit tumor growth in the UCP1 knockout mice.
So without functional heat production in Bt, the anti-cancer effect is lost.
Correct. And the PT scans confirmed it. No increased glucose uptake in the BD of these UCP1 knockout mice when they were cold, and the tumors kept taking up plenty of glucose.
Okay, this is incredibly compelling in mice. Bt needs to be present, it needs to be activated by cold, and its heat-producing function via UCP1 seems essential to create this glucose sink that restricts tumor growth.
That's the picture emerging from the animal models, yes.
Which brings us inevitably to the big question. What about humans? Does any of this translate?
That's always the critical question, and there is some initial tantalizing human data.
Oh, okay. Tell us about that.
There was a small pilot study. First, they took healthy volunteers and exposed them to mild cold, just 22 degrees Celsius, so not the fridge temperature the mice got.
More like a cool room.
For how long? For 7 days.
Yeah.
And using PIT scans, they saw clear activation of brown fat, increased glucose uptake in those typical BE areas around the neck, collarbones, spine.
So mild cold can activate Bt in humans.
It seems so. And then, even more significantly, they did a similar protocol with one patient who had Hodgkin's lymphoma.
Cancer patient. Yeah.
This patient also showed BT activation with that mild cold exposure, but here's the really exciting part.
Yep.
In this patient, the mild cold exposure led to a markedly reduced glucose uptake in their tumor tissue, specifically the mediastinal lymphoma.
Wow. So in a human, mild cold activated Bt and reduced the tumor's glucose uptake.
Exactly. It's just one patient in this report, so we absolutely need larger studies. But it's the first direct hint that the mechanism observed so clearly in mice might actually work in humans too.
That's potentially huge, a
Yeah.
non-toxic way to potentially impact tumor metabolism. Now, you mentioned drugs earlier, mimicking cold. The outline mentions mirabegron.
Yes, mirabegron. It's a drug approved for overactive bladder.
Okay, bit of a different application.
Yes, but its mechanism is relevant. It works by activating the beta-3 adrenergic receptor,
The same receptor norepinephrine hits on brown fat cells when we're cold.
the very same one. So it offers a potential way to activate BA pharmacologically without needing the cold stimulus itself.
Have people looked at it in the context of cancer or metabolism beyond just beat activation?
There's research, including computational studies in silico docking, suggesting mirabegron might interact with other important pathways too,
Such as?
such as AMPK and MTR.
Ah, the big regulators. AMPK, the energy sensor.
Right. Activating AMPK generally boosts energy production, increases glucose metabolism, often seen as beneficial in metabolic health and potentially anti-cancer.
And MTO.
MTR drives cell growth and protein synthesis. It's very often hyperactive in cancer, so inhibiting MTR is a major cancer therapy strategy.
So mirabegron might hit beat MAT in these central metabolic switches.
Potentially. By activating AMPK and maybe inhibiting MTR alongside stimulating UCP1 and BEAT, and even promoting the browning of white fat, it presents an interesting multi-pronged metabolic approach.
Are there downsides?
Yeah.
Side effects?
There are potential concerns. Some studies suggest it can increase blood pressure and heart rate. So caution would definitely be needed, particularly for people with cardiovascular disease. It's not a simple take this pill instead of getting cold solution yet.
Understood. Now, there was also a brief mention about oxygen consumption. How might that relate? We focus heavily on glucose.
Right. The Warburg effect highlights glucose, but it's true that overall mitochondrial metabolism, including oxygen use, is complex in cancer. It's not just about glycolysis.
So altering oxygen use could also impact growth.
Some studies have suggested that yes. For instance, interventions that increase oxygen consumption within tumor cells have sometimes been shown to decrease their growth in vivo.
Interesting. So maybe BT activation, by changing whole body metabolism, could also be affecting tumors via oxygen dynamics, not solely glucose competition.
It's another layer to consider. The systemic metabolic shift induced by beta could have ripple effects beyond just glucose partitioning. It's likely interconnected.
Okay. And one more piece of human data mentioned active brown fat and lymphoma survival.
Yes, another relevant human study. This one looked retrospectively at patients with non-Hodgkin lymphoma who'd had routine PCT scans.
Okay, looking back at existing scans.
Exactly. They checked if these patients had detectable active brown fat visible on those scans.
And did it correlate with anything?
It did. Patients who did have signs of active BAT had significantly longer overall survival and longer progression-free survival, meaning more time before their cancer worsened, compared to patients without detectable active BAT.
Wow. Just having active BAT seemed protective.
That's what the association suggests. And consistent with the mechanism we've been discussing, those patients with active BAT also tended to have lower glucose uptake in their tumors, measured by SUV max on the PET scan.
That really strengthens the case from a human perspective, doesn't it? Active BAT correlating with better outcomes and lower tumor glucose uptake in lymphoma patients.
It certainly adds weight to the hypothesis that metabolically active BAT might indeed deprive cancer cells of resources, impacting their progression.
So wrapping this all up, what does this mean for you, our listener? This deep dive really opens up a potentially, well, groundbreaking path for thinking about cancer therapy, doesn't it?
It really does. Harnessing our own metabolism, specifically through cold exposure or maybe drugs activating brown fat, it's a fascinating concept.
The evidence, from the detailed mouse work to these early human hints, seems to converge on this idea. Shifting glucose away from the tumor and towards heat production in BAT could effectively starve the tumor, or at least slow it down significantly.
It seems plausible, of course, and we have to stress this. Much more rigorous human clinical trials are essential. We're not there yet.
Absolutely. This isn't medical advice, but the potential is exciting. The idea of using something like mild cold or perhaps a targeted drug, maybe alongside conventional treatments.
It offers a potentially less toxic metabolism-focused approach. Integrating these strategies could be a future direction.
Could this lead to more effective combination therapies? Treatments that hit the cancer from multiple angles, including its fuel supply?
That's the hope, and the critical question researchers are likely pursuing now. It's a paradigm shift, thinking about systemic metabolism as a therapeutic target in this way.
It certainly gives us a lot to think about. Thank you for joining us on BioBits and Bytes for this really stimulating exploration.
My pleasure. It's a fascinating area of research.
We definitely encourage you to mull over these findings, maybe look more into how metabolism shapes our health in so many ways. Thanks again for walking us through the science today.